I'm going to talk about trying to expand the repertoire of targets that we consider to be druggable. And as I do that, I'm going to also give you a 20-year perspective on how we've been thinking about translating technologies from our group. And I'll sprinkle in some funny stories along the way. So here's my current disclosure information. Our lab tends to work on commercially projects that have commercial potential, and so we engage different companies early on in the process, and we work with OSAT to grease the wheels there. And I also, on occasion, spin different technologies out of the group. I will talk about some of these companies, but I did want to note that I will talk about IND-enabling and clinical studies related to a compound called KB0742 that is derived from a compound developed in our lab. So if you're unfamiliar with our group from a scientific perspective, we have historically worked on mostly stuff on the left side of the screen here, which is trying to think about using chemistry to explore biological principles. I'll say a lot more about that. We've tried to develop technologies that will enable us to find small molecules that will bind to different types of proteins. That will very much be the focus of the talk today. And as Christina mentioned, we've really tried to think a lot about transcription factors, RNA-binding proteins, cytokines, basically targets that other people consider hard or challenging. We like to think about novel modalities and novel mechanisms for perturbing the function of different drug targets, technology development. And then something that I was not originally interested in but became interested in by coming to the Koch Institute is starting to think about how to apply my skill sets as a chemist or a chemical biologist to drug delivery. And so being in a building with colleagues like Dan Anderson, Paula Hammond, Bob Langer, we've started to collaborate with those folks to think about how we could expand materials for drug and cell delivery, and specifically to develop technologies that would enable us to understand the mechanism of action for materials. So very similar to how we think about small molecules. So I'm going to mostly focus on the early discovery piece. And this is a sort of high-level summary of how we think in our lab. We are not a drug discovery company. We are an academic lab focused on training our students to take their next steps. And so we try to bin our activities in terms of training opportunities. We spend a lot of time surveying different databases, going to meetings, going to conferences, and trying to identify through human disease-guided biology, potential targets that we think would be suitable targets for drug discovery, mostly in oncology, but we do think about other diseases as well. Once we've nominated those targets, we try to assess their tractability from a therapeutic standpoint by finding what we call chemical probes. These are usually small molecules, but they can be other things as well, like peptides. And we want our probes, these small molecules, to physically bind to the proteins encoded by these disease genes. And then we use our probes to understand the consequences of perturbing that protein, ideally in a systems context or a physiologically relevant context. And through that approach, we think we can actually better validate different targets for actual drug discovery in companies. We think we can assess potential novel molecular mechanisms and sort of rule some out and rule some in. And then every once in a while, lightning strikes, and one of our probes is actually a proper lead for development moving forward. And so this is sort of how we think on the chemical biology side. We have biologists, we have systems biologists, we have physician scientists, we have chemists, computational scientists, engineers in the lab that move this forward. And if there's a religion in the lab that you'll see developed over about 25 years, it's that we like to start our process by simply finding pinches of magic dust that will engage that target protein of interest. We're simply looking for binding interactions. So if we run an unbiased binding assay, unbiased with respect to mechanism or the types of agents that we're using, we may find compounds that bind to this target at different sites and they may have different mechanisms. So in a single high throughput screen, we can find compounds that activate our target, inhibit our target. We might find things that cause our target to gain a function, and we might find things that are silent binders that don't actually impact the function of the target, but we can use clever chemistry tricks to convert those silent binders into other types of agents, including agents that deliver our target protein to, say, the proteasome or the lysosome for destruction. So I'll say maybe a little bit about that later. But this is sort of, like I said, a religion in some sense for the lab, that we like to start with binding first. So I'm going to go back to the beginning, though. 25 years ago, I came out to Cambridge to start graduate school at that little red brick house down on the other side of Mass Ave. And I joined this gentleman, Stuart Schreiber's lab, who's in the news quite a bit today because he's launching a new institute that is a biomedical institute in Cambridge that is going to focus on drug discovery and is non-academic. This is a New York Times article that broke shortly before I came here. So anyway, I worked with this dude. And when I joined his lab, which was a synthetic chemistry lab, they were making large chemical libraries on these tiny little 90-micron tentagel beads. And they were using something called split pool combinatorial synthesis, where you put like different things on a bead and then conjugate them, pool them together, split them out again, and develop these libraries. This is sort of old technology. What was transformative in 1998, though, was that they did this not for peptides, but for small molecules. And they developed this library of 2.18 million compounds. And I joined the lab, and I was like, well, what next? So the chemists wanted to make billions of compounds, not millions of compounds. But I wanted to think about, how do you actually explore the utility of these different compounds? So this is me coming in. We didn't have as many robots back in 1998, so we called ourselves grad bots. And my job was to take this pool of beads, millions of beads, and to try and find small molecules from that set that bound to proteins of interest. And so I would take a tube of these beads for that library. I would take my favorite protein, incubate them with that tube, and then I would try to read out a binding interaction using fluorescence. I won't give you all the details, but basically I spent my time over a microscope for hours and hours and hours, taking a little needle and trying to pick out fluorescent beads under a microscope. And my neck really, really hurt, and it was not fun. I had a lot of headaches. So there had to be something better. And the standard in the 1990s was basically to use things like isothermal titration calorimetry, which is a great thermodynamic method to assess binding, or another biophysical method called fluorescence polarization, where you have a labeled, say, small molecule that's rotating rapidly. It binds to a big protein and it starts to rotate slowly. That's a great way to study binding, but both of these methods were not sufficiently throughput to really screen 2 million compounds. So we became interested in spatially addressable systems and took inspiration from this gentleman who was a professor at Stanford at the time, Patrick Brown, who had built these little microarrays of basically DNA microarrays on 3x1 glass microscope slides. And they were printing basically nucleic acids, open reading frames, and they used this to develop some of the first approaches for what we call transcriptional profiling or transcript profiling. So they were mostly following changes in gene expression during yeast sporulation and other biological readouts. And you have to understand that this was all before the sequencing revolution, which is how we do this kind of thing today. But we were really intrigued by that. We actually read his patent. One thing that he did not put in his patent, he said that you could make microarrays of carbohydrates, you could make microarrays of peptides, you could make microarrays of a whole bunch of things. But Stanford did not put the words drug-like small molecules into that patent. So we're like, oh, that's interesting. So not only can we do what we want to do, or we should try to do what we want to do, but there could be an IP opportunity here. And I just want to say, Pat Brown left Stanford to go found Impossible Foods. Okay, so he's probably not mad about the SMM thing. They did actually ask me if I wanted a job at Impossible Foods. So my thesis was basically inspired by Pat Brown's technology and trying to come up with a way to screen small molecules. And so we basically used simple XYZ robots that could pick up picoliter volumes of compounds in DMSO stock solutions and robotically array them onto a series of 3x1 glass microscope slides with special coatings, which I can talk about later. What this results in is anywhere from 10 to 100 micron diameter features on this glass microscope slide. You know, we can make 12 to now many, many more, tens of thousands of features on these arrays. And then we incubate them with a solution of our favorite protein and then illuminate potential putative protein small molecule interactions, again, with fluorescence. So this is sort of the fundamentals. We were not thinking about drug discovery. We were thinking about developing a technology that was low tech and distributable to the wider academic community to enable chemical probe discovery. And I should say we were funded by the National Cancer Institute to develop this technology. So I'll spare you the details on the chemistry. We developed 30 different chemistries. This was actually my first patent. I worked with someone named Hunter Baker, who is someone I work with today. He was not yet in law school. He was sort of in between getting a PhD in chemistry and MD and going to law school. And I still work with him because he was really the first patent, he wasn't quite a lawyer, but a first patent person that I engaged. So I thought this was fun, but actually some of my colleagues in the lab thought there was commercial potential here. I had no interest in 2001 in companies at all. No interest in commercial application of my thesis. But a postdoc in the lab said, hey, there's this thing down the road called the MIT 50K. Why don't we start to work with some of the business school students at Sloan to put together a package? So I kind of came along begrudgingly. Most of the Sloan students, I would say, wanted to be entrepreneurs, but didn't really have a technology or an idea at that stage. And on our side, we were scientists, but we had absolutely no idea about how you would put together a pitch or a compelling package to spin something out. So there was a language gap. And I'll say I was somewhat disinterested in the process. I didn't have a particularly strong opinion of MIT after going through this process, but we made it to the finals. We were one of the finalists. And I remember going into a room. I'd love to know today what the list of people in that room was. But it's mostly non-content experts that were reviewing the finalist teams. And all I could do was just answer questions from a technical perspective. And when we left the room, some gentleman, I have no idea who he is, ran out, took me aside and said, you know what you're talking about. You know what you're talking about, and you're authentic, but you need to know how to pitch. So, but I didn't really care. It's ironic because now what I'll be doing in a couple of weeks is going into a class that I teach, which is one of the only classes here at MIT that's jointly listed with the School of Engineering, the School of Science, and the Sloan School of Management. And the whole point of this class is to get MBA students, scientists, engineers in a room and create that common language and understanding of the opportunities associated with impacting biotechnology. So we've come a long way, but I think quite a bit about that need for common language throughout the institution to make translational impact. So this is something I am excited about, to stick with the theme. So after graduating and going through the 50K, it's now the 100K process, I, again, won't bore you with the details, but I moved to the Broad Institute, specifically 320 Charles, sort of over here, which is an old beer factory, actually. And the first draft of the human genome had been completed, and they were thinking about what next. And the technology that I had developed was obviously a potential technology to help identify tool compounds for all the proteins that we now realized as being part of the genome. So we basically professionalized somewhat and perfected a lot of the surface chemistries associated with that microarray fabrication process, and we really started to validate the technology in a number of ways. So we call it proof of concept to scaled operations. And then in 2006, 2013, was residing in what would become the new Broad Institute building. Started publishing new papers around the platform, new strategies to use the platform, particularly to gain a foothold on targets that have been historically recalcitrant by now not screening purified proteins against these printed arrays, but now screening cell lysates, where you didn't have to purify these challenging targets, which are often inactivated when they're removed from their protein partners. And one really important part, which I was kind of just mentioning, is that we spent a decade basically taking purified protein and screening on the arrays, and we put out lots of databases with our data, because in those days, the idea is you put your hits out in the public domain, they're pre-competitive compounds, the data would be pre-competitive, and it was all what the NCI wanted us to do. But around 2006, we started to change our view on that. And then we also started to explore this lysate technology, as I mentioned. So one really critical thing for us that sort of relates to one of the stories I'm going to tell you about is that when you screen in a cell lysate, your target here in blue, you can find small molecules that bind to your target of interest, but if it's bound to other complex partners, you may find small molecules that bind to those partners as well. And so that's actually really important for a translational story for MIT. And I'll just point that one of my close partners in developing this is Jay Bradner. He's been recently announced as the CSO of Amgen, and he was formerly the president of Novartis Institutes of Biomedical Research. I'll just take you back here and say that's the gentleman in the Red Sox hat right there. All right, so now I was starting to see the commercial potential here. So we were scaled up, we had a robust process. But I was still not, the bug had not bitten me yet. But yet again, I had colleagues at the Broad Institute who had developed a different technology, which was called connectivity map. And there was a lot of interest, particularly from West Coast venture capitalists, in the opportunity to use the language of gene expression profiling to make different connections and insights around drugs. So, but they told my colleagues at the Broad, you can't start a company, though, unless you have a discovery engine baked in, sort of feeding into this other piece. So my friends were like, hey, let's go grab beers. They started to tell me, like, this, we can't launch a company. Venture capitalists won't talk to us unless we bring in the small molecule microarray technology, and we can't do that unless you're on board. And I should say my friends were all non-tenure track faculty. It was a bunch of fellows a bunch of young folks, sort of in our 30s, late 20s. So none of us knew what we were doing. But we decided to go to one of the back rooms of the Black Sheep restaurant about 2-3 times a week and just sort of hash things out. We decided we were going to explore 3 different business models. We were going to explore a contract research organization sort of model, trying to sell kits, and then developing an internal discovery platform. And venture capitalists started saying, hey, why don't you kids come out to Palo Alto, or why don't you come talk to us? One thing that we heard from several venture capitalists is, all I really want is a MYC inhibitor. That stuck in my head. We had to make decisions about, did we want to take a $30 million Series A, which in 2007 was kind of a big deal, or did we want to take on angel funding local, and create a company that's very much us against the world. We see a lot of that at MIT today. We have young scientists and engineers who really want to start a company and they want to own it. I was also told several times you are 10 years ahead of your time, which was frustrating. Made me want to walk away from it all. It was interesting to understand and to gather data about what it was like to start a company while you were fresh into your 30s. We did start a company, and we subleased space from a Langer Lab company called Bind Therapeutics, which was on Sydney Street over here. I moved into apartment in the Kennedy Biscuit Lofts right across the street so that I could run over at night and press buttons on robots. We nailed a partnership with Bristol Myers Squibb, and we hit all of our milestones in that partnership, and it felt great. We spent a lot of time navigating the differences between Bay Area capital and Cambridge capital, and developed a lot of opinions about that. We spent a lot of time with the likes of Peter Thiel's investment groups. And if we do ever grab beers, I will tell you the full story, which would be a lecture on its own. But a funny thing is that there weren't a lot of women in this space at that time. And so I can tell you there were moments where we walked into a pitch and I was the only one wearing a skirt, and I brought my laptop up and everyone assumed that I was not going to be the person speaking. And then you could see the jaws dropped when I was the one who was now going to talk about the science with them. But wearing a wedding ring in Palo Alto, even when you're single, is because no one knows what to do with you, and they immediately start to hit on you if you can talk about science. And then what was really important, and I did not appreciate at the time, was that Palo Alto group had a vision that we did not appreciate in Cambridge, and that was that they realized we could generate large data, lots of screening data in 2007. And they were starting to think about what was more conventional at that time was unsupervised machine learning, but starting to think about how you could couple these data sets to go into deep learning models, which is more conventional today. So I give them credit for that. And then I learned to think about service contracts, whether they were valuable or not. That will become important on the next slide. So 2007 was the worst time to start your first company because of what came in 2008. And while we nailed those partnerships, the funding, and I'm looking at you, Grace, the funding of our colleagues in industry for these smaller partnerships dried up, and the signing authorities changed. And so we no longer had, while we had all of this interest in 2007, by a certain point in 2008, we had no one to continue to interact with. And because of a service contract, because we chose to be cheap, we did not renew a service contract. When we had a catastrophic failure in the lab, we then were faced with the question of, do we shut this down because we don't have enough money to go, or do we try to raise money? In that climate, we made a very quick decision to shut it down. So that's the story of Ligon. I decided to go back to the Broad. Well, I never left the Broad, but to focus on studies at the Broad with that notion that all I want is a MYC inhibitor was a common theme from our Ligon discovery days. So why does everyone want a MYC inhibitor? It turns out that MYC is one of the most prolific oncoproteins in cancer. It's deregulated in 70% of human tumors. We won't go through the slide, but it's involved with all of the known hallmarks of cancer. And so the thinking was that if you could find something that targets MYC, a single agent that targets MYC, you could impact 70% of human tumors. It's a little more challenging. As you're sitting here listening to me, 15% of your gene expression patterns are being regulated by MYC. It has a whole bunch of housekeeping functions. It's important for normal function. If I'm wrestling with my kids at home and one of them scratches me, I need my MYC levels to go up slightly to help with the wound healing process. But in cancer, you have these super physiologic levels of MYC that really drive unrestricted growth and proliferation, giving rise to tumorigenesis. And so what you would really like is not something that blasts MYC away, but that attenuates the amplified levels of MYC that are associated with cancer. So the problem is, why was everyone asking me for a MYC inhibitor? It's because people have been trying this for quite some time in all sectors. MYC is hard because it binds to a lot of different proteins. Most people have focused on this one critical interaction between MYC in purple and a protein in green called MAX. There's a crystal structure of the DNA binding regions of those two proteins shown here. And I'm showing you an MR structure of MYC, which is essentially a piece of spaghetti that rolls into a piece of rotini when it binds its partner. So it's disordered. And as chemists, we don't know how to design compounds to proteins that are intrinsically disordered. So this is hard. It had been a graveyard for drug discovery. And we spent a lot of time in our lab trying to find binders to MYC that would break apart the MYC-MAX interaction. After 10 years of struggling, we decided to take a step back and look at the biology surrounding MYC. There are 3 different MYCs. They bind to this protein MAX, form a heterodimer that activates transcription. And in cancer, you really want to sort of attenuate that signal. MAX can bind to itself, form a homodimer, and that will bind to the same parts of DNA and repress transcription. And moreover, MAX binds to a bunch of other proteins, forms heterodimers that are also repressive. So from a pure academic standpoint, what we were interested in was finding tool compounds, chemical probes, that bound to now MAX and to just explore the biology in this network. We weren't really thinking about drugs. So I won't go through that story in detail, but we worked with a number of different colleagues here at MIT, to screen for MAX, to find novel binders. Some of the work here was supported by the MIT Deshpande Center. The high level is we screened MAX against our microarrays. We found active compounds in cells. We found a compound that stabilized that MAX-MAX homodimer, and it repressed transcription in many cell types. We could demonstrate target engagement in cells. It turned out that this could impact the proliferation of cells. We understood the mechanism of this compound, now KIMS2008, and we worked with a colleague at Stanford to see that this compound actually impacted tumor volume in multiple cancer types. All right, so what do we do with that? Around the same time in the lab, we had another project that came to MYC even though we weren't thinking about MYC. So early on in my time, actually the first 2 weeks of my time as a professor at MIT, I was asked to speak to Janssen about a potential collaborative project as part of an umbrella agreement called the Transcend Agreement that involved the Koch Institute and Janssen. And they were really interested in prostate cancer and specifically finding binders to splice variants in prostate cancer of the androgen receptor. So we decided to use our technology platform to find binders. We jointly developed a critical path where we narrowed it down to a couple of binders that we then worked with someone named Steve Balk at Beth Israel Deaconess to assess in different model systems of prostate cancer. And we found this compound shown here, CARV3, which was this tiny little thing that seemed to be leading to degradation or reduced levels of androgen receptor splice variants. But it looked strange. It looked like a non-specific little compound, maybe a kinase inhibitor. So we profiled it in different kinase assays and found that it was a really selective CDK9 inhibitor. Well, how did that happen? It's because we used that lysate-based screening approach that I mentioned, where we were trying to find things that bound to the androgen receptor or its nearest neighbors. And CDK9 is a nearest neighbor of the androgen receptor. So we continued to study this a bit in the context of prostate cancer, but we knew we now had, with an ultra-selective compound, we had to think about what to do with this compound as well. Moreover, there's this therapeutic hypothesis in the MYC space that if you could find a compound that hit CDK9, you could actually collapse a variety of different feedback networks associated with MYC, because MYC and CDK9 coordinate to drive expression of the androgen receptor and a whole bunch of other transcription factors. But CDK9 is required for the production of MYC itself. So we came back to MYC. So we had 2 compounds of relevance with MYC. We'd basically exceeded our capacity at MIT to advance these compounds any further, because we often work in a one compound, one postdoc model here. So I'll say I like to think about starting a company if you must. This is a line that my colleague Andrew Lowe at Sloan that I teach with uses all the time. As we started to present some of the data around these compounds, not the compounds themselves, my office door at MIT sort of became a revolving door for different venture capitalists. But I want to point out that the team that we went with in the end actually came through the TLO. It was my TLO officer that found the right match for a group that we could create the same vision around integrating the technology platform and bringing in both of these assets into a company to explore all of the utility of all of them. I obviously had great help from the Deshpande Center, and specifically a catalyst, an advisor named Jason Sager, who comes from the pharmaceutical industry. We engaged in connections with which was sort of some of the biggest players in pharma at that time, which is a company called Kite Pharmaceuticals that was acquired by Gilead for $12 billion. So the opposite of the Ligand discovery story, where it was us against the world. This was now us connecting with some of the biggest players in drug discovery. We importantly brought people in from my lab to be the first employees. They used to fight over who was the actual employee. We needed to balance technology development with asset development, and we made sure that we were right off campus at Lab Central so that I could be there all of the time as needed. We brought in a superstar, Norbert Bischofberger, who had been the CSO of Gilead for 20 years, and he's often credited as being the inventor of Tamiflu. And this is a completely different story than Ligand. We had an oversubscribed $105 million Series A, won a bunch of awards. We split the company on 2 different coasts. And the thing that was satisfying here is that Ligand lived on in Kronos. Some of the original vision for that first company that faltered in 2007, 2008 is exactly what Kronos does. And it always makes me think about that person who said you're 10 years ahead of your time. So Kronos started in 2017, and it was one of a generation of companies that were able to sort of rapidly make it to the public markets. We were able, with all of this additional money and additional intellectual capacity, to come in and really build the platform and to advance those assets. And we needed to raise additional money, quite frankly, to move into clinical programs. So they moved on to the public markets October 9th, 2020. During the pandemic, you didn't get to go down and ring the bell. You actually had to do this sort of virtual ringing the bell shown here. I'll just say a little bit more about what Kronos has done with the compounds. They stopped working on one of the compounds that emerged from MIT because there were technical challenges associated with that and returned the IP back to MIT. Another compound, within a fairly short amount of time, they were able to jump from our original compound to a different chemical. That's called KB0742. They've focused on KB0742. In fact, it's a clinical candidate. There's a whole lot of preclinical data I won't bother you with, but the compound clearly impacts MYC-driven cancers of a variety of flavors. It's now in a phase 1/2 clinical trial, and I will just mention a couple of things. It's got a manageable safety profile. They are still in the dose escalation phase, exploring a variety of different dosing schedules. They have a great biomarker where they can see about 50% of reduction in that biomarker leads to an effect in patients. And then just one snapshot that came out recently of some of their first clinical data that's been revealed. They've noticed that the CDK9 inhibitor has been leading to partial response in a couple of myxoid liposarcoma patients. And moving forward, the company wants to explore the utility of a CDK9 inhibitor, not just in myxoid liposarcoma, but other cancers that are driven by fusions of 2 transcription factors, as myxoid liposarcoma is. And then they're also exploring combination opportunities. So fusion proteins, these are often considered to be the most undruggable of undruggable targets. And it turns out that they're often really precise, like ultra-precise targets of interest for pediatric cancers that are very rare, including cancers like pediatric rhabdomyosarcoma, which only impact, say, 500 patients per year. And so as I started to think about what's the most undruggable, tough problem target I can think about, and wow, CDK9 and MYC seems to have an impact, it would be fusion oncoproteins. And then during my time at MIT, the NCI, through the Cancer Moonshot mechanism, created a consortium of different investigators with different backgrounds to try to make an academic impact on these different cancers. So if you're not familiar with what a fusion protein is, here's an example. PAX3 is a transcription factor, and this is what it looks like. FOXO1 is a transcription factor. This is what it looks like. And when a kid is unlucky, these two different proteins come together and fuse and form a fusion that will drive cancer. The other parts are not oncogenic. This is oncogenic. And it's very disordered, very spaghetti-like, so no one has been able to really come up with drugs for these sorts of targets. So we came back to our SMM platform. A number of colleagues here at MIT ran screens, and we identified a bunch of hits, and we've been prosecuting these hits in different ways. I'll just make reference to an ongoing IP story in our lab now. We found a binder to one of those fusions, the PAX3-FOXO fusion. We were able to demonstrate binding and target engagement in cells. And then we converted the compound into what we call a PROTAC. So this is a silent binder. It doesn't impact the function of the fusion, but we could deliver this target now to the proteasome to be degraded. And so this is now, we have filed on this. This is now the starting point for a much larger project in a direction in our lab, something I'm excited about. And to sort of stir the pot, I'll say, I know what to do with a CDK9 inhibitor from a translational perspective, but when you have a drug that could impact, say, 500 patients a year, I can tell you that all of my friends in pharma will not advance that. So we need to think about other types of partnerships, maybe with the government, with different foundations, Summers Way and Emerson Collective. Emerson Collective actually funded us to get some of the preliminary data. And coming back to my PhD advisor in this new institute, maybe we work with some kind of other team to try to bring our probe all the way to a drug for these patients. And that brings me to the Deshpande Center. So another part of this story is that I actually became excited about translation after doing it a couple of times. I benefited greatly from the advice and the funding from the Deshpande Center, which is really here to move technology from labs and to help our colleagues think about how to move them into the marketplace, whether it's through licensing, new venture creation, potentially other models as well. So I stepped into the faculty director role over the summer. I'll just say, we really, moving forward, want to embrace 3 different pillars. It's AI broadly, healthcare broadly, and climate sustainability broadly. And we will likely start to change our process for fundraising around these areas, but also to execute our granting process around these areas. And unlike my experience with the 50K, where you had a bunch of people who might have had some relevant experience, we're likely to create 3 different tracks to support those 3 different pillars within the institute, within the center. There's a company I'm not going to tell you about called 76 Bio that it is not small molecules, but I got to start it with Dane Wittrup. The lesson here was that we had a really cool idea during the pandemic to try to degrade MYC and other targets, not through small molecules, but designed little proteins. The pandemic told us, don't try to deliver the proteins, deliver the mRNA. And the pandemic locked us down, so it forced us to start a company to do the basic work. So this was a miss for MIT on the IP side. It all went to the company. And then finally, what am I really excited about now? What are we working on with the TLO now? We started to think about other undruggable targets, including cytokines. And a few years back, worked with Arturo Vegas at Boston University to find small molecules to hit different cytokines that are traditionally, these binding interactions with the receptors are typically modulated with antibodies. So we were able to use the same SMM technology platform to find things. Arturo's lab has been able to develop better compounds that actually impact tumor growth and to advance them into more potent molecules. But this was proof of concept for us to use our platform in a different way, which was to survey many, many, many different targets for binding and develop binding signatures for now 33 cytokines that we picked intentionally because we knew there was commercial interest. We've had the CEO of Novartis ask for an IL-17A inhibitor. We've had different folks from biotech say, I want an IL-13 inhibitor. So we generated a large data set. We've moved 7 of these cytokines forward into functional assays. This is the kind of data set you get. You get these binding signatures. You can run cheminformatics. You can find selective things. We were very rapidly able to start to identify molecules that we thought would be poised for downstream development through this platform. Here's just one example for IL-17A. And now, yet again, we've sort of, I think, reached our limit for what we can do at MIT. So we're in this place where you must start a company to move things forward. So the new lessons here, we have moved into a different target space, not transcription factors, but we're now thinking about cytokines. We're trying orally bioavailable drugs instead of injection. It's immune instead of oncology. We know that the one postdoc, one molecule model does not work at MIT. And some of you know this will be a very interesting venture because it's starting a company with my husband. So if I come back in a couple years and I'm divorced, you know why. So we will see how this next stage goes, but we are very grateful to the TLO for guidance and thinking through all these novel challenges associated with IP in this kind of domain. All right, so I'll just thank the team. Like I said, lots of chemists, biologists, engineers, scientists. So many people have contributed. What's really great about Boston is the ability to just get on a bus and go across the river and hang out with people. But we have now become more comfortable in the pandemic era with collaborating across the globe. And it's really important to thank patients and families, particularly in the context of the CDK9 story. And then finally, we've benefited greatly from funding from all sorts of sectors. And I have to say, having grown up at Harvard, I know I could get in trouble for saying this, it felt like heretical to think about, actually I would get in trouble if I said the word drug in a group meeting as a graduate student because that was too applied and translational. And MIT has been such a breath of fresh air because now we can think about working with companies almost the same way that we do with just academic partners to try and make an impact on human health. And with that, I'm happy to take any questions.